All right, for this question, we've got to take each individual single-stranded DNA sequence and imagine that it was double-stranded. Now, if you want to do this the hard way, you can actually write in above the sequence or below what the complementary sequence would be, and then you could actually count up all the letters and then try to do the math. But for me, the easier way to do this is to take the 10 letters and actually count how many T's we have. We have 2 T's here. And we also count the number of A's or adenines. 1, 2, 3. We have 3 A's. Remember that for every A you see in this string here, we're actually going to have a T in the other complementary strand. See, 1, 2, 3 T's. So just by counting the number of A's and T's, I've got the total number of T's I would have in the double-stranded molecule, 5. And just remember that I double the number of letters here from 10 to 20. So 5 out of 20 is 1/4 or 25%. I'll do the same thing for the second one. So T, 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 that's 4 T's and 2 A's, that's 6 total. 6 out of 20 is the same thing as 3 out of 10 or 30%. For the next one, I've got 1, 2, 3, 3 A's and T's total. 3 out of 20 letters total would be 15%. And then we got 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 A's and T's out of 20, which would be approximately or exactly 45%.